Our next speaker is Zhen-Qi Liu from McGill University, and following the same concept, he's done a really comprehensive analysis on comparing different types of functional connectivity metrics.
Hey everyone, thank you for being here. I'm Zhen-Qi from Network Neuroscience Lab at the Montreal Neurological Institute. And today I'm going to share with you some of the recent progress on understanding functional connectivity, and that was the previous talk was great. And for this talk, I'm probably going to specifically looking at some of the methodological choices in functional connectivity. So this talk is based on this paper recently published. You can find the relevant code and data on GitHub. And I'll start by asking what is functional connectivity. So most people from my generation probably learned it first as the brain networks, and the brain is somehow acting as a network, and there's the structure and the function networks. However, this first impression might also lead to biases. Unlike structural connectivity, which follows a certain physical tracks or pathways, there's no simple ground truth for functional connectivity. And we construct the functional network entirely from statistical estimation. And the most popular way to do it is probably Pearson's correlation, but does not have to be that. For example, if you're doing EEG or MEG, you're probably more familiar with some spectral measures. And people have been using and simultaneously inventing many different measures. This seminal paper by Stephen Smith is just one of many attempts to study the FT measures people were using at the time. And as a result, the functional networks can be very different. It also poses a challenge that which criteria should we use to evaluate them. These are questions we're trying to answer in this project. So estimating functional connectivity boils down to measuring the relationship between two variables. And this idea is actually more broadly embedded in many scientific disciplines as pairwise interactions. And we use this PySpy toolbox by Ben Fulcher and his group. It allows us to calculate hundreds of pairwise interaction measures, including many commonly used measures such as correlation, distance, mutual information, as well as more obscure measures from information theory, causalities, and so on. So to give you an idea of what measures we use to calculate functional connectivity, I'm showing all the pairwise interaction measures we benchmarked in our paper. The numbers of the measures are the number of variations belonging to that measure. And in total, we have 239 statistics from 49 measures. And in the main results, we calculated all of them for 326 non-rated subjects from HCP Young Adults dataset. Each subject, we have 4 residency rounds, and we're using the shape from 100-node parcellation. So with the 239 FT matrices for each subject, we first ask how similar or different are them. And we quantify this by showing the edgewise similarities between them. What I'm showing here is the similarity matrix with rows and columns being the FT measures. You can see they are highly organized and form clusters that follow their major categories. As an example, the conventional Pearson's correlation is located in the covariance family, which is the top left dark red corner. If we follow it down column-wise, it has a positive correlation with covariance family, negative with precision, distance, then strong positive and negative correlations with some of the information theory measures, and not a lot of correlation with spectral measures. Here I'm showing some of the group average FC matrices just to show the intuition that they're quite different just from appearance. Then naturally we'd imagine they would have different topological and geometric features. So what we do is start by showing the distribution of the edge weights. The 239 measures are side by side on the x-axis. And the darker lines suggest there's a great number of edges. You can see some densities are highly skewed, while others are more evenly distributed, suggesting there's a difference in their topological organization. For example, there'd be differences in network hubs. And here I'm showing the weighted degree or the hubness of the measures. Some measures have typical pattern with hubs in dorsal attention, ventral attention, visual, and some of the model networks. But there is also considerable variability across the measures, with some of them having a more spatially distributed hubs, some emphasizing transmodal regions. Next, we check to what extent each of the pairwise statistics capture two well-studied features of the functional networks. The first is negative correlation with interregional Euclidean distance. And overall, most measures are moderately correlated with distance. There are several measures, however, having a weaker relationship. And the other feature we study is structure-function correspondence. We would typically expect this relatively strong correspondence, relatively strong positive relationship with structural connectivity. Here, we estimated from diffusion MRI. So for most of them, this is true, but there are still some of them having pretty low correspondence. And this suggests even for these well-known properties, there can be a lot of variabilities and strongly depends on the choice of the FC measure. And after these more topological properties, we next consider how they would more directly relate to the underlying biology. And to do this, we quantify how different types of FC correspond to other biologically meaningful interregional networks. Specifically, we have correlated gene expression from the Allen-Human Brand Atlas. We have laminar similarity from big brain cell staining data. We have neurotransmitter receptor similarity from multiple PET tracers, electrophysiological connectivity from resting state MAC data, and finally metabolic connectivity from dynamic FDG PET. We're asking how well each FC matrix aligns with these networks estimated at different spatial and temporal scales. And in general, we observe the strongest correspondence with neurotransmitter receptor similarity and electrophysiological connectivity from MAG. Perhaps counterintuitively, the correlation with metabolic connectivity is not that strong. We also found that across FC measures, covariance precision measures are pretty good, like in general in terms of correspondence, but things are quite different for some information theory and frequency measures. And after studying these more of a group level results, we next turn to some individual level features, and we first test how good they can be used to tear apart individuals, which is a popular analysis known as fingerprinting. We calculated this identifiability index and found that precision, which partial correlation belongs to, really stands out in this one. This is surprising, but also consistent with many previous reports saying how partial correlation might be better at prediction and telling between individuals. And meanwhile, covariance, where Pearson's correlation is, in general, it's pretty robust. We next consider how well these different FC measures can be used for predicting individual differences in cognition and behavior. And for simplicity, we follow the approaches from Tian and colleagues and use this IC-derived 5 components as a target. We then use ridge regression to predict component scores for each individual from the FC measures. Across these cognitive behavioral terms, we see the overall magnitude is consistent with previous reports with some noticeable outliers. It seems like pairwise interactions that perform well for individual fingerprinting, they might also tend to perform well for predicting cognition and behavior. This says it's quite important to select the appropriate FC measure for a specific task, such as this brain behavior prediction here. So now we've seen how FC measures relate to other types of interregional matrices and these external phenotypes. We turn to FC themselves and ask whether we could provide some magnetic explanation. So back to where we wanted to quantify the relationship between two time series, we can think of them as two blobs holding the information they contain. Now to study these two blobs information, one way is to check their overlap, getting the common and unique parts. It's related to the intuition behind mutual information, but this framework we're using is more general. And with this partial information decomposition framework, we can decompose the information between these two variables into 4 atoms. I'm showing here with 4 colors in this Venn diagram on the left. A more intuitive demonstration is on the right. The 4 atoms are unique 1, unique 2, synergy, and redundancy. The unique ones are pretty easy to understand, but what's more interesting are the other 2. So synergy means there are 2 sources of information orchestrated together. They provide new information that cannot be retrieved from either source individually, and I'm here using the yin-yang pattern here to illustrate this. And in contrast, redundancy means each source provides the exact same information as the other, so when combined, they will be repetitive. Now we're going one step beyond this because we're more interested in the information flow between the 2 variables. And back to this Venn diagram, we can replace the labels with the 4 information atoms. And the simplest way to think about information flow is that we give them a past and a future state. Now information transfer happens between past and future. We have in total 16 possible transition patterns. This approach is known as integrated information decomposition, or PHI-ID. The 16 information flow patterns may be intuitively understood as storage, migration, encryption, decryption, and others mapped by this pairwise correspondence matrix. And the theory guaranteeing this to work is quite complex, but there have been many interesting results and insights deriving from this. Dr. Andrea Lupis has some excellent papers. If you want to learn more about this framework, you can also refer to these two original papers about the formal theories behind phi-ID and PID. So here we have these 16 information flow atoms as the matrices. Actually, you might notice the first one, redundancy to redundancy flow here, is quite similar to what we expect from Pearson's correlation. This is indeed the case. And by the way, here's an open source toolbox you can use to calculate these measures. So to see how our FC measures are represented by these information flow patterns, we construct linear models and estimate the contribution of each of the 16 patterns to each FC matrix. So the rows are the 16 patterns, and within each column, the darker or lighter show their relative contribution. We find that for classical statistics such as covariance, precision, and mutual information, etc., they mostly reflect a pattern where redundant information stays as redundant. This is both reassuring and kind of boring in a way. But some spectral statistics predominantly reflecting unique to unique pattern where information that is exclusively provided by one region stays as exclusive to that region. So both cases belong to a pattern of information storage where the information is kept the same over time, but there also exists much more diverse information flow patterns than this. For example, in some information theory and causality measures, we can see the transfer of synergistic information belonging to encryption and decryption, or more formally known as downward and upward causation, if you're more familiar with these terms. And there's actually a lot here. We did not try to interpret each one of them. We're trying to convey the idea that there exists a full spectrum of information flow patterns yet to be explored, and this is not possible with the commonly used Pearson's correlation. And finally, I want to summarize the paper by providing some conclusions and recommendations. In this paper, we see that different statistics give very different FC matrices, and there's substantial variability even for very well-known phenomena. This means FC measure is quite depending on the context. This also alerts us that there is not necessarily a single best measure, but different measures targeting different mechanisms. With no ground truth, it can be very tricky and important to pick an FC measure that will be suitable for your research question. Finally, we have some general recommendations. So what should we do? First, we should always match our measure to the experimental question. In our findings, covariance distance or Pearson's correlation is in general pretty robust, so there is a reason they are used so commonly. We also find precision or partial correlation has some desirable features in some scenarios, such as predicting individual differences. And finally, maybe it's possible to combine or specifically engineer a single or multiple FC measures to achieve some specific sensitivity or precision purposes. And as for future directions, I think first, as you see, there exists a lot of biological complexity in measuring the FC, different modalities, FC methods. They may capture different aspects of it, and there's indeed a lot more to explore. And I really want to recommend this review paper about advancing FC from association to causation. The fact that we see partial correlation stand out in some cases is a great example and also highlighted in this paper. And okay, that's all I have for today. I'd like to thank my lab mates and collaborators. You can read the paper here. Here's how to get in touch with me or lab if you have any questions. And thank you for listening.
All right, we have time for questions.
Me again, hi. Very interesting. Do you think there's any scope for identifying a somewhat canonical subset of metrics so that we all use the same? Do you think that's a possibility, or would there be a trade-off which would be unacceptable maybe for some applications? Or
sorry, what I'm getting is like, do we recommend like using a common subset to
yes.
doing our analysis? I guess
you could do an analysis that you could cluster them, look at the.
Yeah, definitely we can. Like, but it also depends on the research question, right? But like, practically we're already doing like we're already using a subset, which is pretty much pure central correlation and sometimes partial correlation. But we indeed show like they are either in general robust or like better for specific tasks. But like I think the intuition we're trying to say here is like, for example, if you want to study specific information flows, specific correspondence with some of the biological networks, you might need to explore the more like different measures that, or even like engineer some measures to capture this kind of stuff you want to study, like instead of just like sticking to the commonly used measure. So you can adapt based on your research question.
My question, I suppose is about whether there is any theory behind this in the sense that you could identify like a reduced set which is not universal in some way.
Yeah, that's a good question. So in the paper, in the supplementary, we did try to, based on the tasks we performed, we did try to rank them. So we have a ranked list. But basically, in every sense, we cannot say that this is the actual performance that you should basically follow, because we don't have a ground truth idea of what tasks are we using. But from our results, I think I can probably confidently say like Pearson correlation is pretty generally robust. If you still want to use that, that's kind of fine, I guess.
Thank you.
We'll do this. Yeah.
Okay, so my question actually relates to the previous one. Firstly, your talk in the previous one, very interesting empirical analysis of data processing streams and metrics that people use. I think that's extremely valuable. And of course, we've now got computer science type tools to help you do this kind of thing. In the past, I think many of us were complaining about different data streams, but not having a coherent framework to summarize and analyze. So that's great. I look forward to reading the paper. But I want to make a point about theory. So, there is a coherent theory of network construction. The only one that we know, it's called conditional independence graphs, developed in the statistics literature starting in the late 1970s. The classic book is Lauritson's book from the mid-90s. Lauritson was a professor of statistics at Oxford. Partial correlation figures in that, but it's much more general than Gaussian. Related but not really worked out yet is a problem, for example, of autocorrelation in fMRI signals, in EEG signals, in MEG signals, which all those communities have shown themselves to be extremely resistant to taking into account. So I think there are some issues like that, but particularly the conditional independence graphs, which provide a theoretical basis if you have white noise for using partial correlation. So I guess what I would like to suggest, while I think the empirical analysis is useful, you should go and have a look at some of the statistical theory, because I do think there is some theoretical basis that could enable one to provide a reduced set of metrics and criteria to proceed, and while doing that, to have a look at the impact of autocorrelation. There is already some literature dealing with that, but not much, not yet.
Yeah, that's a great point. Thank you for coming.
Well, let's just go for one more question. There's a question on the app, and the person is asking, did you measure conditional mutual information, and are the results similar to partial correlation?
I think yes. So in one of the, we did not specifically interpret each of the measures within the PySpy like toolkit, but all the data and the results are available online, so you can look into them and see specifically one of the measures. If it's similar or different to yours, you can also calculate the one that you would like to use and then compare it to like all of them. So all of them are openly available and online. So both the raw calculated like the PySpy results. So you can definitely like check it on your own. Yeah.
All right. That's all. Thanks, Zhen-Qi, for the great talk.